Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about artificial chromosome vectors. Artificial chromosome vectors are circular or linear vectors that are stably maintained inside cells, and the copy number is usually low, that is 1 or 2. There are different types of artificial chromosome vectors. They are bacterial artificial chromosome vector, P1 derived artificial chromosome vector, yeast artificial chromosome vector, mammalian artificial chromosome vector, and human artificial chromosome vector. Out of these, bacterial artificial chromosome vector and P1 derived artificial chromosome vector are useful for cloning in bacterial cells, whereas yeast artificial chromosome is useful for cloning in yeast. Mammalian artificial chromosome is useful for cloning in mammalian cells, and human artificial chromosome is useful for cloning in mammalian cells. Bacterial artificial chromosome include PBSC108L. This PBSC108L, it was the first bacterial artificial chromosome vector that was developed. Other BSC vectors are PBLO-BSC11, PBSC 3.6. So today we shall discuss about an bacterial artificial chromosome vector known as PBLO-BSC11. This vector contains 7.4 kilobase pair. This is a convenient vector, and selection of recombinant is done by alpha complementation. This vector is stably maintained inside E. coli cells and the copy number is usually 1. By using this vector, we can insert up to 300 kilobase pair of foreign DNA within this vector. Next, we will discuss about various functional modules that are part of this vector. First module is ORES. This ORES is the origin of replication of a plasmid. Second functional module is REPE, REPE. This REPE, it codes for a protein known as rep protein that bind to the ORES region and initiate replication. The third functional modules are par A, par B and par C. Par A, par B and par C are necessary for regular partitioning of BSC during cell division or during division of bacteria. Fourth functional module is there is an antibiotic resistance gene. This is the antibiotic resistance gene known as chloramphenicol resistance gene. So this is the chloramphenicol resistance gene. Fifth functional module is this vector also contains a sequence known as cos-N. This cos-N is the lambda phase cos site. This cos-N is the lambda phase cos site. Next, sixth functional modules. The sixth functional modules are LUX-P. This is the sixth functional module. This is known as LUX-P. This functional module is the site on phase P1 genome. This is the site that is usually present on phase P1 genome. And the last functional modules are large Z. So large Z is another functional module that code for N-terminal segment of the enzyme beta-galactosidase. And other functional modules are T7 and sp6 RNA polymerase driven promoter. There are two promoters are there. This is one promoter. This promoter is bacteriophage T7 RNA polymerase driven promoter, and this is the bacteriophage sp6 RNA polymerase driven promoter. So friends, this is about bacterial artificial chromosome and its various functional modules. This bacterial artificial chromosome can be used to clone up to 300 kilobase pair of DNA insect. Thank you very much. Please share, like and subscribe this video. Bye bye.